I'm going to give a brief overview of the Ficus. My name is Mary Lipton. I work at the Environmental Molecular Sciences Laboratory, and I have a privilege to work with a number of people at our other other partner user facilities to create this Ficus program. So we're going to talk about the Ficus program. I'm going to talk about the Ficus program overall and talk about some of our partners, and then we're going to focus in on the advanced photon source at Argonne. And have a presentation on that facility, as well as a biologist talk about their usage of that facility and how it can be used. And then we'll take questions at the end. So just to go to my next slide, I think we're ready to start. Ficus stands for Facilities Integrating Collaborations for User Science. And basically what it is it's a program started by the U.S. Department of Energy to bring their user facilities together in a way that allows users to come in and access many of the facilities at the same time through the same proposal process. So it allows us to do integrated research without the user needing to have to write multiple user proposals and get them all funded to be able to do multidisciplinary research. The partners that we have within the Ficus right now are shown here. The ones I'm going to talk about today are, of course, EMSL, which is where I'm from, the Joint Genome Institute, the Advanced Photon Source, NEON, and CSMB Biosands at Oak Ridge. So starting just to give a brief introduction of EMSL. EMSL is the Environmental Molecular Sciences Laboratory. We're located in Richland, Washington, and the way that we are organized is we are a user facility funded by the Office of Biological and Environmental Research. We have 150 plus world-class instruments that range everything from microscopy to Raman to mass spectrometry, NMR, XPS, tomography, plant lab. And we basically focus our user program in our science areas. The three science areas we have are environmental transformations and interactions, functional and systems biology, and computing, analytics, and modeling. Just to briefly show a little bit about each one of them, each one of those science areas are broken into what we call integrated research platforms. For instance, the fundamental systems biology is broken down into biomolecular pathways, where we are looking at the underlying pathways in a biological system that translates genotype to phenotype. Structural biology, where we're looking at the structure of the components within a cell, and cell-cell communication, where we're really using our analytics to figure out how cells communicate with each other. Our environmental integrated research platform leads are terrestrial atmospheric processes, where we're looking at that aerosol component. How do aerosols come from plants and soil? How do they perpetuate and how do they degrade? Biogeochemical transformations, where we're looking at what is the biogeochemistry within a system. And rhizosphere function, where we're looking at that plant microbe interaction. And then our computing and analytics has two integrated research platforms, which is systems modeling, where we're focusing on the creation and use of computational models, and our data transformations, which we're streamlining our experimental setups from all the other IRPs to do statistics and visualization and help get data out the door. I'd like to say that any project comes in can use multiple IRPs. We're not stovepiped, but we've organized through the science in this way. Moving to JGI. This is the Joint Genome Institute, which is in Berkeley, it's in Berkeley, California. It's in Lawrence Berkeley National Lab. And their focus is on next generation short read sequencing, as well as long read sequencing, DNA synthesis, and mass spectrometry based metabolomics. So specifically, some of their core sequencing capabilities is de novo sequencing and annotation of a variety of biological systems, including plant, algal, fungal, bacterial, archaeal, viral, comprehensive transcriptomic analysis, microbial community DNA and RNA sequencing, and resequencing for variation detection. There are some custom capabilities within the JGI. There's fluorescence activated cell sorting for targeted metagenomics and single cell genomics, stable isotope enabled metagenomics, as well as whole genome DNA methylation analysis and transcription factor binding analysis for DAP-seq. And then there's enabling capabilities there's metabolomics capabilities where there's an LCM-SMS-based metabolomics analysis of polar and non-polar metabolites for functional genomics, as well as integrated metabolomic and genome analysis. There are DNA synthesis capabilities where you can create genes and pathways through DNA synthesis and then construct whole genome RNA libraries and with QC, and then you can do organism engineering through this. The Center for Structural Molecular Biology is at Oak Ridge National Laboratory, where they can do small angle neutron scattering that provides structural insights into complex biological systems. So we're moving from more of the molecular characterization into the structural characterization. They work with biomacromolecular complexes, biomembranes, and complex systems. They work with a number of isotopes, including hydrogen isotopes, to look at contrast variation in hydrogen deuterium substitution, which is a non-destructive measurement or non-destructive application of the approach to measure the system under physiologically relevant conditions with minimal sample prep. They also operate a deuterium labeling facility that allows for labeling in a number of different systems through fermentation, through HD labeling of proteins, hydrogen deuterium labeling of reagents, and through purification and characterization through their light scattering and their SACs. And then in summary, Bioscience provides unique structural insights into a wide variety of biological systems through target isotope contrast and can be used in combination with other structural tools like light, X-rays, and electrons. And our last user facility that I'm going to talk about is NEON. So NEON is actually operated by the National Science Foundation, and it is a collection of soils and other materials from a number of sites across the US. They have 81 field sites and 180 data products, and you can apply to NEON to get access to these samples. And there's a lot of metadata that goes along with all of these samples. So when you're going to sample, you can go in and collect all the metadata with the weather, the terrain, and everything else that goes along with these samples. And so they have automated instruments that collect a lot of the ground data when they're at their field sites. And then they have sampling capabilities where you can actually get the samples out, as well as airborne remote sensing. So there's, again, a lot of metadata that goes along with these soil samples that can come out of the system. These can be collected on the fly, but they also have a very large archive of these samples. And so the samples are collected through a plot, and they, if it's an initial characterization, they have one per plot. They can do periodic sampling where they have 3 soil cores per sampling event, and then you have a time course or some other course of sampling. And then they also have a plant monitoring zone. And then you can visit NEON. You can visit any of the websites of the user facilities. Before we get into Carolina's talk, I wanted to just briefly remind everybody about the deadlines for Ficus calls. The letter of intent is due March 7th. The full proposal is due March 2nd, and we can take questions on that at the end. And I would like to now introduce Carolina, who will talk about the APS in depth.
Thank you, Mary. Let me get that started. So yeah, thank you, Mary, for the introduction. My name is Karolina Michalska. I am a structural biologist at Argonne National Laboratory, and today I would like to introduce some of the capabilities that APS is contributing to this great initiative, which Ficus is. We joined that program last year, and we are continuing this year. So I will briefly talk about some of tools that we have here. But before I go into that, let me talk a little bit about APS. So for those of you who don't know, Advanced Photon Source is a synchrotron that is operated by Argonne National Laboratory. Geographically, we are located in the Chicago suburbs. It's a national user facility and one of the 5 light sources that are operated by Department of Energy Basic Energy Science. And what APS is, yeah, we have many beamlines for different types of experiments. 68 of them are run specifically by the APS. There are additional beamlines that are run by external consortia. We roughly have more than 5,000 users in a typical year. So it's a substantial user operation at the APS. And what APS is as a synchrotron, it's a very bright X-ray source, roughly 1 billion times brighter than the typical instrumentation that you can find in the doctor's office. And APS until next year had been running for 27 years, I believe. And it was closed last year for a major upgrade. This is a substantial investment worth of $800 million that is going to make that source even brighter, 500 times brighter than it is right now. And with that upgrade, when it's complete, APS will become the brightest source in the US for high energy X-rays. The major component of the upgrade is the redesign of the storage ring, but this is also accompanied by constructions of some new beamlines and enhancements to the existing beamlines. And with the upgrade, what is changing, it's not only brightness that's changing, also coherence is increasing, and this all will bring new opportunities, especially at the imaging beamlines, because we will be able to probe larger samples at very high resolution and also much higher speeds, which is very suitable for biological and environmental samples where heterogeneity is a substantial problem, and many samples need to be tested to actually get some understanding of these environments. And also with the upgrade, some of the time resolve experiments will become feasible, for example, at the macromolecular level, where we will be able to monitor reactions, catalytic reactions as they happen in the protein crystals, for example, to learn some more about catalysis at the molecular level. So all these upgrade features are really highly relevant to biological and environmental science. And we really hope that that scientific community that is pursuing the research in these areas could really utilize these resources to their advantage. And the IPS is still being closed to users, but will be coming back online in spring. The first call for new proposals will open in March 4th for the summer run. So there is a possibility for users to apply for some beam time for the summer period. And APS runs a general user program as a national user facility where everyone can apply for the beamline, but we just started a new initiative that we call eBerlite, and this particular program is targeting specifically scientists that are working within the biological and environmental science. And that program provides a comprehensive platform for the types of experiment that we envision might be run, but this kind of community. And we also expect the user community may not be overly familiar with synchrotrons, so we are here to help navigate APS ecosystem and help designing experiments and lead the researchers throughout the entire process, from planning the experiments through execution up to data analysis. And through that platform, we are providing support for 5 X-ray methods. The primary techniques that we are supporting is crystallography, X-ray tomography, and X-ray microscopy. We have some effort as well in the X-ray absorption spectroscopy or scattering. And to be able to properly do all these experiments, we are also providing laboratory infrastructure for molecular biology and also spaces for sample preparation, such as cryo laboratory for specimen preparation under cryo conditions, or infrastructure that requires anoxic conditions, for example, for environmental samples. And there is also a huge computing resources to support data processing that is coming from the APS. This is the entire Eberlite program, but through the Ficus, we specifically providing crystallography, tomography, microscopy, and laboratory resources for sample preparations. And I will talk a little bit about these different techniques and how you can use them. So I mentioned microscopy, and specifically through Ficus, we will be doing X-ray fluorescence microscopy. This is a scanning microscopy where you use small beam and you scan your sample, and each point in your sample, you collect X-ray fluorescence spectrum. And this is a very sensitive and also selective method, allowing you to identify elements that are present in a given space, because a spectrum is really very specific to each element. And at the APS, we can detect all elements that are heavier than aluminum, so we can access most of the biologically relevant elements here. It is especially good for trace elements here at the APS. These experiments can be done in 2 dimensions. So this is 2D elemental mapping, and there is also a possibility to do this in 3 dimensions to get a volume distribution of specific elements. And the way this technique, or the applications of this technique, extends to study physiology of microorganisms. One can look into aerosols chemistry, biogeochemistry, and, for example, plant biology, where we can look into elemental absorption, response to toxic metals. We can test growth conditions, or looking into roots behavior and visualization of such tissues. We currently have 6 instruments available through the Eberlite program that utilize this technique. 2 of them are brand new beamlines that are currently being constructed. And additional beamline, BioNanoProp, is undergoing some enhancement as well. And this is one of the beamlines particularly suited for biological work, because it's the one that enables to test samples under cryogenic conditions. So this one is being upgraded to increase resolution from 13 nanometers to 10 nanometers. And collectively, all these instruments allow to cover a broad range of resolutions across length scales that are relevant to the investigated system. So we can address microorganisms or aerosol particles at nanometer resolution, and we can then also be looking at larger plant or soil samples at lower resolutions, covering a pretty broad range of scientific questions. And I have a few examples how these can be utilized. For example, this is an X-ray fluorescence experiment that was done by Joseph Jake's lab, who is investigating wood decay and the role of fungi in these processes, or more specifically, the role of ions in fungal degradation. So in that experiment, the wood chips were exposed to soil, and they were inoculated with 3 strains of fungi that are known to belong to different classes of fungi. The experiment was done at 2 different instruments to look at different resolutions for these samples. And you can see here that different species have different capacity to absorb elements from soil, and that helps them to participate in this lignocellulose degradation. The major difference that you observe here is the Serpula strain can absorb iron, and the Serpula, as long as together with Glauhelium trabium, these are the brown rat fungi that are known to utilize iron in Photon reactions used to generate free radicals that down the road help to depolymerize woods. So and the last organism is not able to, or is not able to absorb iron, probably because it doesn't really need that for physiology. And the next example here is the work published by Tamás Varga and co-workers, and here they were looking into phosphorus solubilization in plant tissues. Again, this study involved many different techniques, and X-ray fluorescence was one of them, to look how this phosphorus migrates to the plant cells. And phosphorus is one of these essential elements for plants, but its availability is somewhat limited as the majority of that element in the soil exists in soluble forms, not that easily absorbed by the plants, and the plants require participation of microorganisms to help solubilize phosphorus and absorb that. So the experiment was looking into samples that were inoculated with the bacterial strains that are known to help with the processes, and that was tested against control samples that didn't see these strains, and there was marked difference in distribution of phosphorus in inoculated in not oculated in control samples, proving that phosphorus uptake was indeed supported by the symbiotic strains. And not only that, the presence of these strains also affected the morphology and biomass of the root, and this was shown by complementary imaging technique, computed tomography, which was looking into the structure, morphological features of the roots. And tomography is another technique that we offer through the Ficus program. Instead of in the tomography experiment, instead of scanning through the samples, we are using full field imaging. So the sample is exposed all at once, and you add rotation, and that allows you to reconstruct 3D image of your sample to probe structural features of that object. We have 3 instruments available at Eberleit. Although I should make a note that APS as a whole has more beamlines with such capabilities, but those are the ones that we offer through the program. So again, there are 3 of them, and they vary in their experimental envelopes, allowing to study either larger samples at lower resolution or small samples with very high resolution to really focus on areas of very special interest in a given system under study. So I'm showing here 3 examples how this can be used, and 2 these images are coming from collaboration with Andrei Smirtenko from Washington State University. He investigates genetics of embolism resistance in grasses. So we are looking here at the structures of grass samples, which were subjected to drought, and we are monitoring changes that occur in morphology of certain cells under drought conditions. We can then quantify behavior or presence of certain features in these samples. And again, the experiment was done at 2 different beamlines and at 2 different resolutions and sizes of the samples. And the same technique can be used to study soil structure and to some extent also content, soil porosity. This is a very important tool for environmental scientists. Many of such researchers are looking into soil structure. And again, this can be done at with different sizes and different resolutions. I'm showing just one example here. This is collaboration with Ken Kemner. In this particular experiment, we were testing applicability of one of the beamlines to study really large soil cores, because we know that environmental community really is interested in this large scale behavior of their samples. And another example I have here, this is from Georgi Zbabnik work. Georgi is looking into the interactions in the poplar rhizosphere. So they collect the poplar roots from across several states, and then they image them through micro CT tomography, and they later apply AI machine learning tools to analyze the data, look at the characterization of soil particles and porosity and the distribution of these pores in the samples. And again, the research itself is not only about imaging, it's also connected to genetics and phenotyping of the strains associated with the samples. And then last but not least, we of course supporting crystallography techniques to determine 3-dimensional structures of proteins, protein-protein complexes, protein-DNA complexes, protein small molecule complexes, substrates, products, cofactors, everything that comes to mind. We have 3 beamlines to use. Again, not the only ones at the APS, but those are the ones that we will be utilizing in the program at Sector sector 21. We can do traditional crystallography, which is based on the single crystal under cryo conditions. So entire data set is collected from a single crystal to get a structure, just a single snapshot of that structure. Or we are now venturing into what's known serial crystallography or dynamic crystallography. These types of experiments are done on thousands of crystals. And the premise there is that you just collect a single image from a single crystal, and then you move to the next place on your sample, on your grid, shoot another crystal, and then you keep doing this until you shoot enough crystals to obtain a complete data set to determine one structure. But this is done at room temperature, so it allows you to access more physiologically relevant temperatures than the cryocrystallography. And this tool, what it also allows us is to look into time-resolved experiments. We can then probe, or we can time the data collection with the initiation of the reaction. And this experiment allows to monitor catalytic reactions in the crystal. We just need to properly design the triggering of the reaction, so we can properly determine the timing of the data collection with the reaction stage that we are interested in. And on the right, there is an example of the proof of concept experiments from Andrzej Joachimiak's group. In that experiment, we were looking into reaction catalyzed by the beta-lactamase enzyme. What this enzyme does is hydrolyzes beta-lactams that are present in a certain class of antibiotics. It's a metal-dependent enzyme. So we were able to obtain crystals that didn't have metals, and we were monitoring how these metals the zinc ions were released to the crystal. And with the proper delay of data collection, we were able to capture certain stages of the reaction. And with this, you can sort of generate this kind of movies where you can see how the reaction is progressing. And we are hoping that with the APSU, we will be able to do more experiments of this kind to help with the research in catalysis in general. And to support these types of experiment, we have a protein production pipeline and advanced protein characterization facility. This has been developed over many years, and it covers all necessary steps in the process. We have robotic gene cloning. Over the years, we developed more than 120 plasmids for heterologous expressions of proteins. The pipeline has the protein expression system to grow bacteria, yeast, or insect cells for expression. We have also mammalian cells for that. Semi-automated protein purification. We can characterize proteins enzymatically. We can study ligand binding and other types of interactions when needed. Then we have semi-automated crystallization and imaging, and we have, again, beamlines next door to collect diffraction data on these crystals. This facility operates as one of the sectors at the APS. It's called Sector 84. So again, everybody who wants to use APS can apply and use these resources, come on site and do the experiments themselves. But we also have some capacity for mail-in crystallography, where the potential users can just submit sequences of interest, and we can do the work for them. And in addition to that, we have extra spaces for sample preparation. I mentioned earlier a well-equipped cryolab for sample or tissues manipulations. And in addition to that, we have anoxic chambers, walk-in freezers, and plant growth chambers. We learned that many experiments actually require plants to be grown on site. They need to be close to the beamline, so the sample doesn't suffer through transport, and we actually can test it just close to the conditions where we wouldn't want to study them. And I think this is all I have. We obviously encourage everybody who would like to work with us and develop proposals for Ficus to contact us before, and we can help with some planning and designing experiments and telling what's feasible, what's not feasible. So you can contact myself, mostly for structural biology, or contact Zoe Finfrog, she is on the call as well, mostly for imaging and other resources at the APS. And I'll finish here. Thank you. And next, I think we have Sabiha Merchant from University California, Berkeley, who is an APS user, and she's going to tell you about her science that she's able to do here with us.
I am Sabiha Merchant, and I am a biochemist and plant and microbial biologist at the University of California in Berkeley, and I'm also a faculty scientist at LBNL. So what I'd like to do today is tell you about some of the work that we have done using the resources at various user facilities to understand copper and zinc metabolism in green algae. So first, I'd like to tell you about Chlamydomonas. On the left-hand side, you will see various images of Chlamydomonas. These cells are about 8 to 10 microns in size. And this is relevant because we'll be talking about spatial resolution when we try to look at elements subsequently. And of course, each cell has a particular elemental profile, which you can see here on this log scale with macronutrients making up the biomass, and then the trace elements allowing various types of catalysis to occur. In my group, we have optimized methods to drop out one nutrient or one element at a time to understand how the cell handles deficiencies in essential elements and how the genome responds to such deficiencies. So as you can see in the bottom set of bar graphs, that we can deplete cells of macronutrients so that they grow on a more minimal quota, such as for nitrogen deficiency. And you can also see that we can grow them on minimal amounts of iron, manganese, zinc, or copper. So today I'll be focusing on our work on copper and zinc deficiency. So just the take-home message from what we have learned from studying copper deficiency at the level of the transcriptome and proteome is that hundreds of genes are upregulated in copper deficiency or downregulated in wild-type Chlamydomonas cells. And you can see this here in this heat map. We have a whole set of genes highlighted here in yellow, whose expression goes up in copper deficiency. The expression of these genes is dependent on a transcription factor called CRR1. And you can see that it's the CRR1 mutant, these genes are not regulated anymore by copper. I'm just going to talk about a few genes. So I've expanded the heat map here so you can look at some genes that I'll be referring to. For example, the CTR genes that encode copper transporters. CYC6 is a cytochrome that covers the function of an essential copper protein in photosynthesis. And CYC6 is a heme protein. It's our biomarker for copper status of the cells. And then here you see coprogen oxidase, which catalyzes a rate-limiting step in heme biosynthesis, which we also use as a biomarker. And each of these genes is upregulated in the wild type in copper deficiency. And in work that we have done collaboratively with MZEL, including Mary Lipton's group, we have essentially shown that all the transcripts that are upregulated in copper deficiency end up resulting in changes in protein abundance. So essentially, the changes in the RNA drive changes in the corresponding proteins. And here you can see the increase in abundance of the cytochrome C6 protein. Now, the operation of the signaling circuit ensures that the right amount of copper is found in cells. So if we look at the graph on the left-hand side here, we see that when we give the cells very low amounts of copper, barely detectable amounts of copper, they take it up with high affinity through these CTR transporters that are encoded by the genes that I just mentioned. However, once you give the cells the copper that they need, you can give them 100 times more copper and they still do not take it up. And this is in part because the genes are turned off, but we believe also in part because the proteins themselves are degraded. And we are studying this degradation process in collaborative experiments with MZEL using proteomic methodologies. So on the right-hand side, you can see what I mean by regulation of the CTR transporters. Here we have CTR1, 2, and 3, and you see that in plus copper cells, this is qPCR data. There's barely any expression, but in minus copper cells shown by the fill circles, you see that the expression is much higher. All right, so now I showed you that the copper content of cells is very tightly regulated. When we move to study zinc deficiency, and we always measure the elemental profile of our cells by ICP mass spectrometry, we saw that in zinc deficiency, of course, in low zinc shown in the green bars, we have much less zinc in the cells. However, when we looked at the copper content of these same cells, we noticed much higher copper levels. And remember, I showed you on the previous slide that the copper content of cells is tightly regulated in wild-type cells. But this is only under zinc replete conditions. When you drop zinc out of the medium, the cells hyperaccumulate copper. So this was a puzzle to us. Yet, when then we looked at the biomarkers to understand the copper status of the cells, what we saw shown on the right-hand side in this bar graph is that the CYC6 gene, which remember I told you was a biomarker for copper status, in zinc-deficient cells, the CYC6 gene was massively upregulated. Note that the y-axis is broken here. So it's, very dramatically upregulated. Same thing with the CTR1 gene. This is the copper transporter. I showed you that they are regulated by copper. And again, in zinc deficient cells, although there is a lot of copper in the cells and the medium is replete for copper, the CTR transcript is also increased. And in fact, the entire copper regulon is increased. So this presented a conundrum in the sense that we had chemically overload of copper, but biologically we had deficiency. So how did we address this question? We were interested to understand why the copper was invisible biologically. So we took advantage then of the user facilities at ANL using the advanced Photon source. We used the bio-nanoprobe there with the help of their beamline scientist, C. Chen, and learned how to do X-ray fluorescence microscopy. So in this particular experiment, I believe we're looking at cryopreserved cells. So here is a single chlamydomonas cell. Remember, it's about 8 to 10 microns in size. And then we can image either iron or copper or a number of elements. In this particular case, we're looking at the chemical or physical properties of the elements themselves. And so there's no ambiguity about which element you're detecting. So we're looking at fluorescence emissions that are element specific. So there's a hard X-ray source. I should tell you that what we're doing here is we're simply scanning the cells with a short dwell time and 70 by 70 nanometer probe so that we can cover a single cell in about 3 to 5 hours. And here you can see foci of copper and iron in these cells. So this is one kind of technique that you can use to look at elemental distribution in cells. In parallel to this, we also used a different technique. And again, this relies on the chemical property of the element itself. In this case, we're looking at the mass of the element. And of course, copper would have a different mass than, say, iron or calcium. And again, here is our chlamydomonas cell. In this case, these cells are fixed and sectioned like you would for transmission EM. And you can see again that we see foci of copper, which are co-localized with iron. So this suggested to us that copper and iron were co-localized in cells. We also took advantage of a chemical probe that had been made for copper by Chris Chang's lab. And here you can see that this BODIPI-based sensor can bind copper out here. And if we stain these zinc-deficient cells with this fluorescent probe, again, you see hotspots of copper. A control dye, which has carbon in place of the sulfur, of course, gives us no fluorescence at all. And it was important for us to use all three methods because, of course, the fluorescent copper probe that can always be subject to artifacts because we're looking at the fluorescence of hydrophobic molecule, which of course depends on the environment that the molecule is in, whether it's aqueous or hydrophobic. But these two chemical methods are very powerful because they can unambiguously show us or can resolve individual elements. And the advantage of the X-ray fluorescence, of course, is the cryopreservation capability that we used in this particular project. So this explained to us the conundrum of why copper was invisible biologically, and the reason is because it was sequestered in a compartment that houses these metals. So the next question we had is, how is copper found in this compartmentalized situation? So in this case, we used another user facility, and that was spectroscopy at SLAC, again, using an X-ray source. And what I'm showing you here is a near-edge spectrum, X-ray near-edge spectrum from 3 independent preparations. And you can see it's highly reproducible because all you get is one line. And this is consistent with copper-1 species. I'm not going to show you the XAF spectrum that we did, but that told us that this copper may be bound to sulfur and nitrogen ligands. So our next question was, what might be these ligands that are associated with copper in the cell? And here we took advantage of metabolomics capabilities at JGI with Trent Northern's group. And of course, our initial thought was that maybe this might be glutathione that's binding copper, but in fact, it turned out to be cysteine. And you can see here that in zinc deficient cells, we have huge content of free cysteine amino acid. And when we went back and looked at our ICP-MS data, we noted that, in fact, we had a statistically significant increase in the cysteine content of zinc-deficient cells. So at this point then, Stefan Schmolinger in the group thought, well, we should be able to go back and look at our X-ray fluorescence data from the advanced photon source and ask the question whether the excess sulfur in the cells is co-localized with the copper. And you can see that result here. So again, we're looking at 5 independent cells, 1, 2, 3, 4, 5. But let's just focus on the one in the middle. And these are cells that were preserved either by cryopreservation or by chemical fixation. We get the same results. And what you see is that in low zinc, we have co-localization of copper with sulfur. Of course, the sulfur signal is not as sharp because sulfur is present in many proteins and molecules in the cell. So we always have a background of sulfur, but still you can see the foci of concentrated sulfur, and which is co-localized with copper. And in the merge, of course, you can see this co-localization clearly. So finally, because we had established that the fluorescent probe that Chris Chang made would identify the same hotspots of copper that we could detect chemically and physically, either with nanosims or with X-ray fluorescence microscopy, we could now use this chemical probe to look at the dynamics of these copper storage compartments. So here, I'm going to show you an experiment where we ask the question whether copper can be mobilized from these intracellular foci. And in the top panel, we see our zinc-deficient cells. And again, it's very evident that in all of the cells, we see these foci of copper. When we add zinc back to the cells, we restore copper homeostasis, and the copper leaves the organelle. So the question we had is, which of the copper transporters is responsible for the copper leaving the hotspot? And what you see here, this is the wild type cell. In 7 hours, all the copper has left, so you see no staining. And we noticed that only in the CTR2 mutant would you see that the copper in these foci remains within the foci and does not leave. So this told us that the boundary membrane of the compartment where the copper is localized has CTR2 type of transporters, and these transporters are responsible for efflux of the copper from the cells. So let me just end here and tell you that what we showed in this work is that zinc is required for normal copper homeostasis. We knew from metabolomics that this excess copper was sequestered with cysteine in a lysosome-related organelle. And the sequestered copper can be mobilized, and for that, we need the CTR2 transporter. And the important part with respect to the Ficus projects is that we used multiple different techniques. For example, we can use resources at JGI for the transcriptomics experiments, MZEL resources for proteomics. We use JGI resources for the metabolite analysis, namely the sulfur-containing amino acids. And then finally, we used various beamline and synchrotron facilities, such as the advanced photon source, for localizing the elements within the cells with high spatial resolution, and then also resources at SLAC for understanding chemical speciation. So I'll thank you for your attention and acknowledge the various co-workers. I'll particularly point out C. Chen at the Advanced Photon Source, and at JGI, Yun Tao Hu and Trent Northern, who helped with the metabolomics. At PNNL, it would be Mary Lipton and Sam Purvine. So thank you again.
Let's see, we have a question for you, Mary, if you want to come back on. Crystal Zunga in the chat asked a couple questions. Can neon perform, can they perform changes into the neon sampling points? So I think that might be for Emzel.
It would be for neon. And I'm not sure how they set their sampling up, whether it's you can go in and request a certain sampling site. Michael San Clemens, who I will put his contact information in the chat, would be the person to answer for this. And I'll be putting the contact information for the people from each of the user facilities in the chat, so that you'll have access to them, and potentially put a slide up that has all of them at the end as well.
She also asked, can you perform suggested treatments in the neon sampling points? So that might have been the same question asked a different way.
Yeah, I don't know that they treat in the field. So, I don't know whether they're doing temperature studies where they have the temperature cones. I do know that with the neon samples, if you get a core in, we can do microcosm experiments that you can treat those samples in different ways. So I think treating in the field is rather difficult for them. I think they're set up to just go sample, but we can certainly do after harvest treatments to look at physiological changes.
Okay. Carolina and Krista, if you want, and Zoo, if you want to come back on camera, we've got a couple other questions that can be answered. So one, and any of you can take this if you'd like, who is eligible to submit a Ficus proposal? Krista, do you want to take this question?
Sure. Eligibility is very open. So international, US, different career level stages, it's pretty much okay with us, as long as it's okay with your institution. I think some institutions do prevent postdocs from being PIs, but that would be on your own institutional level. I don't know if any of the other user facilities have different restrictions.
So let's see, David is asking, what are the acceptable budget categories for the Ficus Awards?
I'll take that one. It's kind of hard to answer because every facility is different. And the amount that each facility can spend on these awards is different. So my suggestion is you don't put a budget together, you request facilities. And so my suggestion is when you're putting your LOI together to reach out to the people in each of the facilities, and I put the contact information in the chat, so you have it. Reach out to the contacts that are on the and they can direct you to the right person that can get your project in the right scope for each facility.
And then another question, what can I ask for as part of the Ficus call? I would say, Carolina, if you want to take that for APS.
For the capabilities that we mentioned, so the request is for capabilities for a given experiment. So the crystallography, the full field imaging, the microscopy, and the use of laboratory resources. Also separately, I think for plant growth chamber, if we want to plan, because the capacity for this is a bit limited, so we have to look into how many applications we are getting and balance balance between different applications, what can be done. So essentially it's a request for a beam time, if you will. No, it's not a funding request, it's the request of the time on an instrument or in the laboratory. No, does that answer the question?
And that's the same for all the facilities. You are not asking for money. We will not be sending money to your organization. You are requesting access to capabilities, instrumentation, and analytics at each of the facilities.
David is, oh, sorry.
At no cost to you.
Yes.
Right. And along that too, David is asking, what about travel? Do you want to take that, Krista, since you're on?
So since there's no money exchanging hands, travel is not something that any of the user facilities would cover.
Okay, and then a couple more questions. How can I maximize my chance for success?
Talk to us ahead of time. Reach out and talk to somebody at each of the user facilities you want to use. And don't go in blind. We are here to help. And like I said, I listed the contact information. I'm not going to tell you about microscopy. If you contact me, I'm going to forward it to Scott Lee, who is our expert. So we have a myriad of very talented scientists who do this, and they are a wealth of information that you can lean on to create a very strong proposal.
And I guess we could add that it's important that the science is within BER mission, right? Because that applies for everything that we are talking about here.
Absolutely. Yeah. Any words of wisdom, Krista?
I was just going to add the DOE mission as well, but I guess also a plea to not wait until the day before the deadline, because you'll get a better response if we have a little time to think about things with you.
Absolutely.
We should add too that while it needs to be within BER mission, it's not required to be currently funded by BER, right?
And BER mission means we're not going to do human, we're not going to do human gut microbiomes. We're not going to do tumor cells or cancer cells, anything that's funded primarily by another agency. But if what you want to do is a sea sediment microbial community that answers the call and answers, this is the community I have, it fits into, I can use it as a model system that fits to answer something in the call, you can still propose that. But if it is on a system that is way outside of DOE mission, it's going to get rejected.
Yeah. Carolina or Zoo, how about how will the upgraded beam lines at APS improve capabilities for Ficus proposers?
So we want to take that one.
I think Carolina already covered those in her talk.
Mhm.
It's really, for biological environmental system, you have to survey fair number of samples that to answer a question. So it's really the speed of collecting data, and also with the increase of coherence, so we can really have a high spatial resolution. You can resolve feature that was not able before. It's just, that's all my thing. The advantage is the speed and the spatial resolution.
Oh, that's great. And Fabian is asking if we're going to share the slides from Sabia, and yes, we will. You'll get that in an email follow-up. And with that, is there any other questions from the audience? Okay, going once, going twice. All right, we're not seeing any more questions. So thank you everyone for attending today. We hope that this has been enlightening for you to learn a little bit more about Ficus. If you have any questions, you are always welcome to reach out to any of us that were here online, and we can either answer your question or direct you to the correct person. The JGI Ficus call, the JGI Emsl Ficus call that is open right now, the letters of intent are due in about a month. So you have a little time to, as we said, reach out now and talk to folks to get your proposals in the best shape possible. So with that, I think we will say thank you and have a great day. So, all right.
Thank
Bye,
you,
everyone.
Thank you.
Thank you.
Bye.
Bye.